So firstly, I'd like us to consider how our industry is structured today. There are organizations available to you as development scientists that are configured to produce API, develop formulations, and manufacture clinical trial supplies that all exist in a vertically organized channel. However, these companies are very separate to CRO test type organizations which exist in a separate channel. And this means that a lot of time is required to transition a drug product from the make channel to the test site. Because of this separation, there's an increase in the CMC data package required, obviously an increase in drug substance consumption, cost, and then there are further impacts associated with that on the overall development time. If we now consider what this structure means for formulation development, we find that the process described on this slide has emerged. A pharmaceutical development program is performed in the make channel to identify formulation prototypes, which are then screened in a preclinical species, usually dog. Successful formulations can then be subjected to conventional CMC analysis to generate an extensive data set to support a regulatory application for a clinical study. This process can take a long time and carries considerable cost, with independent assessments of between 12 to 18 months and $1.5 million to $2.5 million of outsourced cost. As a result of this time and cost, a lot of emphasis becomes placed upon laboratory and preclinical assessments, but these are widely accepted as not being predictive of human performance. This in turn increases the risk of having to repeat this overall development cycle more than once because the correct product is not identified first time. At the heart of this problem is the fact that our supply chains allow entry into clinic only once these conventional CMC processes have been conducted. So what if we could integrate our industry horizontally to improve this? That's exactly what we've done at Quotient. In our facility, we have an 85-bed clinic on the ground floor, and right upstairs from that is a fully functional lab/pilot-scale GMP plant. Outside of this, we're supported by formulation development and pharmaceutical analysis functions, as well as data analysis and regulatory groups. This platform of translational pharmaceutics means that we can manufacture a batch of products upstairs in our facility, move it downstairs and dose into a patient, in some cases in less than 24 hours. Fundamentally, the furthest a product needs to travel between production and dosing in our facility is around 200 feet, which is a very short distance indeed. The translational pharmaceutics platform therefore allows us to enter clinical dosing with a shorter shelf life on our product, and therefore we need a reduced CMC package to enable us to get that study off the ground. We would typically submit data to support a 7-day shelf life, which will remove ICH stability studies from the critical path. They can instead be run in parallel whilst that PK data is being obtained. In addition to this, manufacturing happens at small scale, so development of larger scale production processes can also be removed from the critical path. Finally, because cycle times between manufacturing and dosing are so short, we can enable rapid make and test cycles. This means that real-time decisions can be made on the basis of emerging safety, PK, and PD human data that crucially allows drug products from one dosing period to influence the drug product that is manufactured and dosed in the next period on a 7 to 14-day cycle. So this translational pharmaceutics platform now allows us to reconfigure drug product screening processes to enter the clinic more quickly and perform rapid manufacturing and test cycles. Now, successful products from the Pharmaceutical Development Program can be identified and regulatory approval can be achieved on batch analysis and short-term stability data collected on demonstration batches of products. Once regulatory approval is achieved, we can select which product we wish to dose first, make that formulation and dose it, before cycling back with that data to decide which prototype is studied next. This can further be enhanced by the application of formulation design space, where key formulation parameters can be modified freely to optimize a drug product. This concept in particular has been a major innovation within formulation development processes, and later in today's presentation I'll show the breadth of product parameters and also the degrees of flexibility that we have applied to date. So as a result of the changes to the development paradigm, RapidFACT can reduce development time with clinical data emerging from as little as 4 months into a development program. Flexibility and precision is enhanced through the application of make and test cycles and of also formulation design space. One key benefit that can be delivered is in around API consumption, which is greatly reduced because we only manufacture drug product we need to support the immediate dosing period and not prepare large batches to support extensive stability studies without knowing that that product is going to proceed into later development. All of these benefits can be delivered irrespective of whether Quotient conducts the development process or whether this happens at a sponsor or a third-party CDMO. But fundamentally, there is an increased probability of achieving the right drug product